Hi, welcome to Unit 5. I just want to go through some of the more important slides in this PowerPoint just to explain any of the more difficult concepts, etc. So this is our guy of the PowerPoint. His name is Joseph Edward Murray, and he was the plastic surgeon who is famous for completing the first human kidney transplant. He did it on identical twins. Now, can you think why that was cheating a little bit? Have a think. That's where we're going in the textbook. Having a look at those pages. So remember what that means. It means that if there's anything asked from the textbook, this is what's going to be asked. We're not using the colouring book at present, so don't pay any attention to that. So the urinary system, just as to introduce it, is obviously the system that makes the urine, okay, and it's part of homeostasis. We produce urine for many reasons: getting rid of waste products, adjusting the concentration of the urine, and also it's something to do with blood pressure as well, which you might not have thought about. But in this slideshow, we're going to have a look at the two different, the male and the female systems, which are pretty much the same actually, and we'll get on with that now. So you can see there where the kidneys are. This describes where they are. I don't think you should find this in the least bit difficult to understand, so I'm moving on with that one. This again describes the shape and the different parts. Superior poles, inferior poles, and that sort of thing. Again, should be pretty easy for you. Internal anatomy of the kidney. Now, let me just go through this. The kidney is bean-shaped. Kidney bean-shaped, to be exact. It has an artery and a vein. The artery supplies blood to the kidney, and the vein drains the blood away. And you can see that on the outside of the kidney, there is a fibrous capsule. Then there is this layer here above these pyramid things called the cortex, and the pyramid things are in the medulla, and the columns go between the pyramids. Okay, and they're actually called pyramids. Then we have a minor and major calyces. These little dips. If this was an actual kidney or a model of a kidney, these would be indents. This is where urine drains out of the ends of these pyramids and gathers in these calyces. So first into a minor calyx, and then into a major calyx, and then it drains into the renal pelvis, which is this area here, and then it drains down this tube called the ureter, and that goes to the bladder. Okay. Here it is in it will be in a little bit more detail here. I'm sure you can read about that. And here is an actual kidney compared with the diagram. And if you were to have a lab and dissect a kidney, it's one of the things that actually looks like it's meant to when you dissect it. Unlike the heart, when you try to dissect a heart that's being pickled, it's very difficult to find out which way round everything is. So you need to know all these parts for the exam. And again, there it is. The hilum is just the bit where everything goes in. If you recall, there was a hilum in the lymphatic system in the notes there. Okay. So it talks about the renal medulla here and the pyramids, and the pointy bit on the pyramids is the apex, and they're full of small tubes called tubules. A tubule is a small tube, and we're going to look at those because the functional part of the kidneys is in the pyramids, and we'll look at some detail with that. So the renal pelvis is funnel shaped, and it basically drains the kidney into the ureter. And you have a major calyces and you have minor calyces, and they vary in number depending on the person. So all sorts of different variations. And here we're just showing the location. Notice that the right-hand side kidney is lower than the left-hand side kidney. It's pushed down by the liver. Here are the ureters, one on each side, one for each kidney. This is the urinary bladder. So when you produce urine, it drips down this tube, which is one of the longest tubes in the body. It's a foot long, and eventually gets into the bladder. Okay. And looking down here, you can see where the kidneys are in the cross-section of the body. And again, you can see that the right-hand side hangs lower. Do you remember what HCO3 is? It's a bicarbonate ion. It's to do with maintaining the pH of the blood, which we'll see in some detail when we do the physiology part. But I just wanted you to see that there. So, functions of kidneys: maintains and adjusts the blood plasma volume. And here's the first inkling that it's to do with blood pressure. The more blood you have, in other words, and blood's a mixture of stuff, including mostly water, like everything else. The more water that you have in the blood, the higher the pressure, because you don't grow any more tubes. So it's like having more water in the same tube, so the pressure on the tube is greater. Okay. We remove waste products, and we adjust the concentration of electrolytes. We can get rid of sodium, etc., and helps maintain plasma pH. So remember, I think when we did when we did the heart, we said that the pH of the blood is 7.35 to 7.45. So the kidneys are very important in maintaining that pH. So basically, we're going to look at here that the internal part of the pyramid is called the parenchyma, and inside this substance are nephrons, and the nephrons, which look like this, and there is like 1 million of them in each kidney. This is where your urine is formed. Okay, here's the blood, and coming out at the end here is going to be urine, and in between it's filtrate. So once it comes out of this blob here, which is called the glomerulus, it drips into this tube here, and it's filtrate because it's been filtered from the blood. And it travels along this set of bendy or convoluted tubules, down this big long loop of Henle and up again, and some more convoluted tubules and into something called the collecting duct. And once it gets into the collecting duct, it's pretty much done. It's urine. Certainly once it drips out of the end of the pyramid, it's urine. Okay, so the ureters, as we said, are these tubes. They're up to a foot long, and they have connective tissue inside them and smooth muscle which can push stuff along. This is the best I could do, I'm afraid, but you can see it's coming out of the kidney and into the bladder. So the kidneys are bilateral. That means, quite simply, because there's one on each side of the body. So the bladder is a hollow bag, essentially. It's in the pelvis, held in place by the peritoneum, and it's lined with transitional epithelial tissue, which you'll remember can change its shape. So it can get flattened and hold more urine. And it's surrounded by a detrusor muscle. And that detrusor muscle is what you can use to force the urine out. And there are two rings of muscle called internal and external sphincter muscles, and they are the muscles that you relax in order to allow the urine to go out. And you have a certain degree of control over it. Not when you're first born, but you develop control over it. And so that you can essentially choose when and where to get rid of your urine. Obviously, it can't go on forever. Something's got to give sometime. So the urethra is then the one single tube that leaves the bladder and goes to the outside world. In females, it's about 4 centimeters long, and in males, about 20 centimeters long. Essentially, in females, it's a very quick exit out through the vulva. In males, it has to go along the length of the penis, so it's a much longer journey. And this also explains why bladder infections are more common in females than in males, because the bacteria causing a bladder infection have further to travel in males to get to the bladder. Okay, so this again explains where everything is. We've just gone through that, and you will have read it. This is just recaps pretty much what you've got, what you'll read when you're actually going through this PowerPoint in detail. So we're going to look at the structure of a nephron now. Remember, these are the little things, there's about 1 million of them in each kidney, and they make the urine. It's made out of a renal corpuscle and a renal tubule, which we saw. Renal corpuscle is this area here, and the tubule is all the rest of it. And that's the collecting duct at the end. So, two types. There are cortical ones, which by the name suggests they're in the cortex mostly, and juxtamedullary. Juxta means near, so they're near the medulla. And in fact, the juxtamedullary nephrons, their loop of Henle goes much further down. And there are two parts, the corpuscle and the tubule. So let's have a look at them. This is a standard one. You can see that they are actually inside the pyramids, and they look like this. They have a blood supply, and the blood supply goes through this network you can just about see in there, which is a network of capillaries. And the pressure in there forces the liquid part of the blood out, the plasma. A lot of the plasma comes out with a lot of dissolved chemicals. And one of the main jobs of the tubule is to reabsorb a lot of the stuff that we still need. We produce way too much urine, filtrate I should say, and if we produce that amount of urine, we'd essentially be drinking all day whilst peeing all day, which obviously we don't do. So then we have this loop of Henle coming down. Its purpose will be explained. It's to do with water reabsorption, and then some more convolutions, and then going into the collecting duct. Okay, this is a juxtamedullary nephron. You can see it's much longer. This is the dividing line between the cortex, the outer layer, and the medulla, the inner layer. And with the juxtamedullary nephron, it goes way further down than the cortical nephron. And it also has this series of blood vessels called the vasa recta around it. Okay, so cortical nephrons are mostly in the cortex, short loop of Henle, and make up 85% of all nephrons. Whereas the juxtamedullary ones are found much closer to the medulla, and they make only 15%, and they have very long loops of Henle, and they have the vasa recta as well. So we have the glomerulus, which is that cluster of capillaries, and the arterioles that go into and out of the Bowman's capsule or the glomerulus. So you can see here what's happening is that we have an afferent arteriole which brings the blood into this glomerulus, and the glomerulus is the filter. And so a lot of the solid parts of the blood remain inside the blood, and a lot of the liquid comes out through little holes. It's just like any other filter. It's got holes in it, and the size of the hole dictates what can come out. And then you have your efferent arteriole. So unlike a lot of other things where you have a venule coming out of a capillary network, with the glomerulus, you have the arteriole comes out. So you have the afferent going in and the efferent going out. And note the relative size of these two. This is fatter than this one because a lot of the liquid has left. So you have less blood leaving than you have coming in. Okay? So the blood pressure is higher in the glomerular capillaries because we need to push the liquid part out. We need a higher blood pressure to force the filtration. It's a bit like an espresso machine, if you know how that works. Efferent means to leave. Proximal means closest to. Okay, so here you have it in a little bit more detail. Your afferent arteriole, bringing the blood in. The filtrate is made because there's high pressure inside this network here, pushing the liquid part of the blood out, and then the rest of it goes back into the efferent arteriole. The glomerular capsule is wrapped around the glomerulus. It's also called a Bowman's capsule, and it's there to collect the liquid, the filtrate that's coming out, and then it's carried away down the proximal convoluted tubule. Okay. So it's a site of filtration. There's the Bowman's capsule, the glomerular capsule, and you've seen what that does. It has a parietal layer around the outside, just like any other parietal layer, and a visceral layer that wraps around the capillaries themselves. Okay, and there's the capsular space which collects the filtrate, and it then goes off to the proximal convoluted tubule. So we look at the tubule. The tubule is narrow and it carries filtrate. It's a site of reabsorption. Remember, we make too much filtrate. We have to take some stuff back in, and that really mostly happens in the proximal convoluted tubule, the first part of the tubule. Reabsorption is what goes on. We do get some secretion because even though we filter out a lot of the components, we don't filter out enough of some of the waste products, so we have to actually actively push them into the filtrate, and we have special cells to do that. Okay, so we have here, just to do the route, the afferent coming in. You have the pressure here creating the filtrate through the little pores in the glomerulus, and you have the efferent vessel taking the rest of the blood away. The filtrate travels through the proximal convoluted tubule into the descending limb of the loop of Henle, up the ascending limb of the loop of Henle, through the distal convoluted tubule, and then into the collecting duct, and then finally dripping out of the end of the renal pyramids. Okay, so proximal convoluted tubule, the loop of Henle, distal convoluted tubule, and collecting duct are all parts of the tube, the renal tubule. And we're going to look at them independently here very quickly. So the proximal is the first part. You can read about it there. The loop of Henle is the second part. What it does, it adjusts water, reabsorption of water. The distal one is the third part, and it basically it's there, and then the final part is the collecting duct, which receives the filtrate. It's almost urine then. The water content and salt content and hydrogen ion content can be adjusted in the collecting duct, but once it's been adjusted, it's urine, and it remains the same from the time it leaves the end of the renal pyramid till the time you pee it out. Okay, so we have around the renal tubular, which means around, tubular means around the tubule, capillaries around the tubule. And they come from the efferent arteriole, and they wrap themselves around the tubule. They don't go down the loops of Henle. All nephrons have them. Then we have another blood vessel series, the vasorecta, only in juxtamedullary nephrons. They go all the way down the loop of Henle. And only descending portion of the vasorecta, on the descending portion of the vasorecta, sodium, chloride and urea are reabsorbed and water is secreted. Okay, and on the bit that's going back up again, sodium and urine are secreted and water is reabsorbed. So you can see what's going on later on. We'll talk about it. We've done that before. If we look at the nephron's histology, the membrane, we have the endothelial capsular membrane. That's where the blood passes through. That's your filter. Okay, and it's a combination of the visceral layer of the Bowman's capsule and the endothelium of the glomerular capillaries. So it's the structure of the capillaries and the structure of the visceral layer that cover the capillaries that work together to make the filter. It has endothelial basement membrane sandwiched between the two of them. So you can see here it's showing what the things are made out of. The glomerular capsule parietal layer is made of a thin layer of cells there, epithelial cells. And here you've got more structure going on in this basement membrane and podocyte. This is where filtration happens. We have this thing called a fenestrated endothelium. Fenestrations mean windows or pores. And then in the proximal convoluted tubule, you've got a much tighter membrane, and the same with the distal convoluted tubule. And your nephron loops are different. Now, you're not going to be asked about the different types of cells, so I wouldn't worry too much about that. But it tells you about them here. Important ones are here, the fenestrations, the windows, which allow stuff to pass through. They're not really windows. They don't have any glass in them, but they're holes, like windows. Okay. Then you have the basement membrane, etc. So you have these things called podocytes, false feet, or types of feet that basically are like the teeth in the comb, and they can be adjusted to adjust how much fluid is let out. Okay, because between them are these slit pores or filtration slits. So if you narrow the slit, you reduce the amount of fluid being released. So they restrict passage of medium-sized proteins, so smaller components of blood can pass through. You don't want the big bits of the blood. You don't want your formed elements leaking out everywhere. That's not good. In the distal convoluted tubule and the collecting duct, the epithelial is simple cuboidal. Remember that? No microvilli, but there are two important types of cuboidal cells present. We have principal cells, which produce, they respond to antidiuretic hormone and another hormone called aldosterone, which we'll mention when we do the physiology. And intercalated discs, which cells, that secrete hydrogen ions. Hydrogen ions, if you're secreting them, you're increasing the pH of the blood, because you're getting rid of acidity. Okay, so if you can adjust the pH of the blood by adjusting how many hydrogen ions you're secreting. Okay, so there they all are again. So this table here tells you what's filtered or reabsorbed. So here, substances filtered into the Bowman capsule are all these things. So this is what comes out of the Bowman's capsule into the collecting duct. Glucose, amino acids. It only, at first glance, you can see this is stuff we need. So we better take it back in. And look at the first one, glucose. Think about diabetics. The test for your blood sugar levels and to see if you're a diabetic originally was actually to taste the urine. People would taste the urine to see if it was sweet. Then it moved on to peeing on a stick that would change colour, because it had an indicator on it that would change colour in the presence of glucose. Now we have much more accurate and sort of like instantaneous readouts with blood sugar meters, etc. But the point of this is, if you don't reabsorb your glucose, you're diabetic. That's what they look for, and the levels in your blood. So a lot of these things we want back, so we have to reabsorption. We have reabsorption that will take place. So this will tell you what's reabsorbed. So in a healthy person, all your glucose is taken back in. All your amino acids and lactic acid is taken back in. And then the rest in proportion here, which you should really pay attention to. In the loop of Henle, 15% of the water is reabsorbed. And in the ascending limb, so that's in the descending limb. In the ascending limb, sodium ions, potassium ions, and chloride ions, to those extents, are reabsorbed. Okay, what is secreted, in other words, what we pour into the tubules actively, in the proximal convoluted tubule, hydrogen ions, ammonia, which is one of the breakdown products of urea. It starts off as being urea. Creatinine, which is from muscles, and they do measure that, and urea from the breakdown of amino acids. And urea again is secreted in the loop of Henle. In the collecting duct, hydrogen and potassium ions can be secreted. And if you secrete your hydrogen ions, you basically cause filtrate and urine pH to decrease. You make the urine more acidic and the blood less acidic. Now get your head around that one. A decrease in pH means it becomes more acidic. An increase in pH becomes less acidic and more basic. Okay, and some potassium ions to ensure proper heart function. Don't eat more than 1 banana a day. Okay, so here we talk about the blood supply to the kidneys. 20 to 25% of the whole cardiac output at any one time. And these are the arteries that it travels down. Now, we're not really going to do much about these, but you would if you did the real lab. But we start with the aorta, then we have the renal artery going into the kidney, which then becomes the segmental artery, the interlobar artery, the arcuate artery, and the cortical radiate artery. Then associated with the nephron here, we have the afferent arteriole, the glomerulus, and then the efferent arteriole. It's not a venule. And then we get the peritubular capillaries of the vasa recta, which feed back into the cortical radiate vein, the arcuate vein, the interlobar vein, the renal vein, and then the inferior vena cava back in the heart again. Okay. So there they all are, very pretty, and we would, if you were in class, we would do a beautiful lab that shows all those. Micturition is peeing, basically. And it's controlled by, first of all, you need to know when you want to go to a pee, because stretch receptors send messages to the brain saying, hey, we better pee soon. And you go, oh, I better pee soon. And then we get an involuntary action, which we don't control, that relaxes the internal urethra sphincter muscle and the detrusor muscle, which starts to contract. So there's even more pressure on you to go and have a pee. Eventually you go, well, I better have a pee now. And you go to the bathroom and you release your external urethral sphincter muscle and the pee comes out. Simple as that. Normal urine, we produce 1 to 2 litres per day. Its colour is clear to yellow amber. It's not cloudy normally. Its pH is between 4.6 and 8, depending on what we eat. And the specific gravity varies from 1.01 to 1.035. That means that it's denser than water and will sink in water. And it's normally 95% water and 5% dissolved salts.